All right, so for this video, we're going to be looking at D2.3, which is focusing on water potential. We're going to be going with the theme of continuity and change. So how is water going to change as a byproduct of solute concentrations? And we're thinking about what factors affect the movement of water into and out of our cells, and how is that going to be similar or different in plant and animal cells. So when we're talking about solute concentrations, we're looking at tonicity. And so we have hypertonic, isotonic, and hypotonic. And so solutes in a liquid, in a solution, are going to affect the bonding between water molecules, and that's going to make it harder for water to move. So water is going to move from an area of low solute concentration to an area of high concentration. And we learned this when we looked at what osmosis is. And so we have three different types of, these are all red blood cells, and they could be in a hypertonic solution. So that means there's more solute in the solution than in the red blood cell. And if that's the case, the water is going to move out and these red blood cells are going to shrink, and eventually they might plasmolyze, meaning they kind of collapse in on themselves. If they're in an isotonic solution, that means it's equal between the solute concentration between the red blood cells and the solution. So water is going to move in and out, but there's not going to be any net change to the red blood cells. And then hypotonic is going to be when there is a lower concentration of solutes in the solution. And so water is going to move into the red blood cells and cause them to burst. And so we would want, if we're talking about medical procedures, to be using isotonic solutions. So our eye drops that we would put in our eyes, or IVs of rehydration fluids or something like that, are going to be isotonic solutions. So water movement in plants, water is going to move, this is true in animal cells as well, but it's going to occur due to osmosis. Plasma membranes are selectively permeable, so they can change the speed of osmosis by changing the permeability of the plasma membrane. And they do this by indicating that they need aquaporins, so there's channels for water to move through the membrane more easily. Plant and animal cells have different structures. So animal cells can easily burst in hypotonic conditions because they only have a thin plasma membrane that is easily torn. Plant cells have a cell wall, and as a byproduct of that, that cell wall is made out of cellulose, which is much thicker, and it has a high tensile strength, almost more than steel. And so what that does, that tensile strength allows plants to oppose gravity, but it also allows there to be a lot of pressure on the inside of those cells. So the cells can fill up very, very, very full with water and press on the cell walls without bursting. And a cell that is full like that is said to be turgid. Plasmolysis, however, is when the water in the cell decreases sufficiently that the membrane pulls away from the cell wall. And so you could see here, this would be plasmolysis inside the cell of a plant. So to summarize, isotonic solutions, the water is going to move in and out, but no net change is going to occur to the cell because the concentration of the solution is the same as the concentration of inside and outside the cell. Hypertonic solutions are going to result in a net movement of water outside of the cell because there's more solute outside than inside, and it's going to end up having that cell become plasmolyzed. And in hypotonic conditions where there's less solute in the solution, the water will move in. And in the case of plant cells, those cells will become turgid. In the case of animal cells, they might rupture. And all of that water movement is due to osmosis. Plasmolysis is going to be where the membrane is going to pull away from the cell wall, or where the cell just collapses in on itself, like we saw with the red blood cells. The next slide is about HL. So if you're SL, you can stop here. HL student, we're going to look at water potential and how it causes osmosis, and that's going to be affected by temperature, solute concentration, and pressure. So water potential is going to be triggering movement of water, and movement of water is going to always occur from a hypotonic to a hypertonic solution because of the water potential. And PSI here, this little trident symbol is the symbol for water potential. So water potential is 0 kilopascals at 20 degrees Celsius at 1 atmosphere for pure water. And so if you had pure water on both sides of this tube, then there would be no net movement of that water. But if we increase the solute concentration, then what's going to happen is that is going to lower the water potential of the solute, and it's going to cause the water to move towards the solute on the right side of the tube. Additionally, if we added pressure, so here we can see we're adding positive pressure onto the left side, which is going to increase the pressure water potential, the water potential pressure, and that's going to cause the water to move to the right. If we created a negative pressure on the left side, that's going to mean that the water is going to move towards the negative pressure. So if we increase solute concentration or hydrostatic pressure, that's going to decrease water potential. And water is always going to move from the higher pressure or potential to the lower potential. So in the case of it being 0 here and it's, let's say, -10 here, then it's going to move towards the solute concentration. If you bathe a cell with a potential of -400 kilopascals in a solution of -300 kilopascals, then the water is going to move from the solution into the cell. And that's because the cell here has a more negative potential than the solution does. And that is all for this unit, this section.